So when working in the lab, you must wear full PPE, and that must be a lab coat and latex gloves. Each lab practical will have a risk assessment and COSH paperwork, which includes the chemicals in use. All this paperwork is found online, and if you ever have any doubts, the lab practicals are supervised by members of lecturing staff, and you must ask questions if you ever have any doubts or any questions you may need answering. In today's tutorial, we're going to be using the AP test. This test looks for and helps to detect and identify seminal fluid on exhibits such as underwear, intimate swabs, for example. There's an enzyme in seminal fluid called acid phosphatase, and that acid phosphatase is very high volumes in seminal fluid. The test we use is a liquid test called acid phosphatase AP test, and in the presence of the enzyme, it produces a colour change. The first step is to ensure that your AP reagent is working correctly and in order. So we do a positive control, i.e. we have some fabric here with a known seminal fluid deposited on there, and we have a negative control, which is the same fabric but with just water. To do this, you get a piece of just normal white filter paper and you place this over your fabric, and you can mark the edges of your filter paper just so you know which orientation that paper has been placed. Using sterile water, you want to spray on top of the filter paper so it soaks right through to the fabric. Again, try not to oversaturate it, but kind of do the coverage, the total surface area. And you might want to do this with your negative control. And again, apply a liberal amount of water. So there's no volume, you just keep spraying until it kind of coverages it. Again, get some just normal acetate paper, place on top, and with your gloved hand, press firmly for about 4 to 5 seconds. And then you want to remove the filter paper and not the fabric, and then this test needs to be then taken to the fume cabinet, and that's where we spray the AP onto our filter paper. Once we've got the filter paper in the fume cabinet, this gives us an opportunity then to actually spray our AP reagent onto our filter paper. The AP reagent is kind of like a straw-like solution, and we always spray it into like a spray bottle. The reaction has a 2-minute reaction time. So when we spray it in a second, and we spray 2 or 3 pumps of the spray over our filter paper, if we've got a positive control and a positive result, it must have to change colour within 2 minutes. The colour reaction is from a colourless, i.e. a colourless stain, and if it goes a purple reaction within 2-minute time period, we deem that as a positive AP reaction. So we get the filter spray bottle and we spray 2 sprays, and we then look at our results. Now we're going to spray our AP reagent on our controls, so just nice 2 or 3 reactions, and we then set our timer for 2 minutes, and we sit back and just see if we actually see a reaction within a 2-minute period. Once our controls have passed, we're now able to enter the exhibit bag and retrieve our exhibit to examine. So we always open the bag on a different seal, and we simply cut our exhibit bag with a pair of scissors. And with a latex gloved hand, retrieve your item, place onto your examination desk. You then want to open out your garment. In this case is some black underwear, and given the case circumstances of the crime, In this case, we are doing a targeted search to see if we can identify semen on the inside of this underwear. So you may have to turn your garment inside out and place it onto your examination paper. Using your examination note-take paperwork, you want to do some detailed note-taking on the description, condition, and any labels, any logos, or any visible staining. Please use some white light to see if you can see any suspicious or kind of white staining, any blood staining, before we carry out this test. And this is an opportunity to recover any hairs, fibres onto the underwear before we carry out the AP test. The next step is to get some just chromatography filter paper and place this filter paper on an area of interest. So this time we're doing the front inside surface. So you place your item. You may want to use a ballpoint pen to draw around your item so you know the edges of your garment, so you know the actual surface the filter paper has had made contact. Using sterile water, you want to liberally, again, do not try and overspray to oversaturate the item, but this might take 20 seconds to just kind of spray, just sterilise water onto your item. And once you've covered the total surface area, you want to get yourself a acetate plastic sheet and place this sheet on top of the filter paper, and using your gloved hands you want to make some pressure, i.e. press the acetate, thus pushing the filter paper onto your item and making contact. Spend about 5 seconds doing this, and then once this is done you want to peel back the filter paper including the acetate sheet, and then this has to now go, like we did with the controls, into the fume cabinet. That's where we will spray the filter paper with acid phosphatase AP reagent. So once we've brought the filter paper back to the fume cabinet, we then get our AP reagent, we spray it to get the total surface area of our filter paper, and then we have a 2-minute reaction time. So we stand here and observe for up to 2 minutes, and any reaction which goes from a colourless to a purple reaction, like our positive control, we then have to document it on our notes. It's good to keep an eye on the time period. So as we can see here, approximately 20 seconds in towards the reaction, we are obtaining a purple reaction. We still leave it for the full 2 minutes to keep an eye on the intensity of this purple reaction, but that started to change within about a 20-second time frame, and that information is really important to put on your examination notes. And we stand here observing until the full 2 minutes, just to see if any other areas do come up within that 2-minute time zone. Once this is done for the 2 minutes, we then take the filter paper sandwiched between 2 acetates back to our exhibit, and then this reaction, this filter paper, helps us to localise where there could be visible sperm cells within the acid phosphatase positive areas. So we have to place an acetate sheet over, so you kind of sandwich the filter paper between 2 pieces. The reason for doing this is when we take it back to the exhibit and place this back onto our exhibit, that none of the AP reagent makes contact with our exhibit. Because AP, if it gets into the reaction, would not be very good for the DNA profiling process. It will inhibit the reaction. So once we've come back from the fume cabinet and we have our AP paper, which we have a positive reaction here, we need to place this, the clean acetate, down over our garments, over our underwear here, and you want to kind of adjust it to make sure that paper is in the exact orientation that it was previously. This is very important because any slight deviations could not be where we will actually target that semen. So on this case, you then peel it back, including the acetate sheet, and using like a contrasting colour, so this is a black fabric, so yellow shows up really well. You want to kind of pull back your reaction like this and carefully draw around where the purple AP positive reaction is. So afterwards you can take this to place it to one side for now, and within this yellow circle here we have a positive semen sample, and within that fabric and the fibres of that material, if it's sperm cells present, we now need to recover it. So from the whole garment here we have localised an area of interest. We then cut or swab these areas, and we then do a slide and we look through a microscope to see if we can actually see sperm cells. So once we've localised an area and we've marked up on the exhibit, we're then able to use just a wet and dry swabbing technique to recover that body fluid, that semen, from this area. So we simply use a wet and dry swab. So first, a wet swab dampened with DNA sterile water, and pressing firmly, use the total surface area of the swab and place the wet swab into your swab case, and then quickly, before the water evaporates, use a dry swab, follow up with the same area and place the dry swab in the swab case. These two swabs are now ready to go forward to make a microscope slide and to be able to see what's on there. So the next step in the procedure is, once we've recovered the AP positive staining from the underwear, we've now put them on swabs. So now we need to convert like what's on here onto a microscope slide. Because remember, the AP reaction is only a presumptive test, doesn't confirm that there's semen present. So we have to visualise to see if we can see sperm cells from these swabs. And the only way to do that is using a histology slide, and to see if we can actually see visible sperm cells on the slide. So we have the two swabs, and what we have to do is each swab has to go into the same, these are called Eppendorf tubes, which are 1.5 mil tubes. So we open our lid and we get our swab, and because it's got a plastic component to the swab, we have to carefully, using scissors, chop the swab head off. So there's the wet swab, and in the same tube we put the dry swab in there, and exactly the same process. We use a pair of scissors, be very careful, health and safety, that you chop the plastic off. So there we have the two swabs in the Eppendorf tubes. So that's great. Next step is we have to put 1 mil of water, sterilised water, from here into our swabs in our Eppendorf. And to do that we need to use a pipette. Make sure that you adjust it to 1,000 microlitres, which is 1 mil. Get the corresponding tips and unscrew your sterilised water. Press down, take up your water and transfer that to our Eppendorf. So what we've done there, we have now 1 mil, dispense of your tip safely, we have 1 mil of water which has submerged our swabs. And the next step now, we need to beat them cells off them cotton buds, and the only way to do that is to use equipment called a vortexer, and that's what we should do now. The next step is to use a vortex, and the principle is it's a vibrating machine, and you simply just press your sample onto this rubber head and it vibrates, and the theory goes is the cells which are on our cotton buds need to be vibrated and to go into solution. So you simply make sure the lid is closed, press down, and it'll speed up and you do that for 30 seconds. Once your 30 seconds is passed, you'll see that your white swabs would have discharged the cells, and your solution then will have the cells in solution. Ready for the next step is to put a small proportion of that onto a slide. The next step is to remove these two swab heads from this Eppendorf into this Eppendorf. So open your lids and using a clean, sterile tweezers, nice firm hold from here to here, and then your second swab head into here. And then close your lids, and as you can see we've moved it from here to here, and you have this solution left here, and this is what we're going to make the slide from, a very small proportion of this, and that's what's going on our microscope slide. The next step is, once we've removed our swab heads and we have this Eppendorf with our solution in there, we need to spin it down to make the sperm cells go to the bottom, to make a very concentrated, what we call a pellet of cells. To do this, we need to use equipment called a microcentrifuge. So make sure everything is on, switched on first, open the lid by pressing the button, undoing the rotor lid a few times, which will release the lid. And inside here you have a thing called a rotor, and in there you have different size holes which will take different size tubes. In this case we've got these Eppendorf 1.5mm tubes. Place it in one of the holes, but we never put a sample in with just one tube. We have to balance the rotor, otherwise it doesn't spin round perfectly. It'll make a really loud droning noise in the lab, and sometimes it can be unsafe. So it's important that you balance the centrifuge. To balance it, you need a corresponding Eppendorf, preferably with the same amount of solution, and just use water, but you'll find them around the lab if not. Place it right across, so balance it right across from each other. If you've got two samples, you'll need two balances, etc. And once they're nicely in the holes, get the rotor, turn it clockwise until you can't turn it anymore, and then using your hands just nicely close the lid. On the dial of a centrifuge, you can change the speed of a centrifuge. For this procedure, we need it to go 13,000 rotations per minute for a time period of 30 seconds. You can adjust the speed and the time using the dials appropriately. Once you have your time and speed set, simply press green for go, start, and you'll hear the centrifuge build up speed up to your desired, in this case, 13,000 rotations per minute. And once your 3 minutes is over, you will hear your centrifuge slowly go decelerate, and once it stops moving you'll hear a beeping noise, which then allows you then to open the lid. Never open the lid until you hear the beeping noise, and then press the open lid, anti-clockwise open your rotor lid, remove the lid, find your sample, and if your centrifuge has worked properly, you will see at the bottom of your Eppendorf a very white, kind of opaque pellet of cells. And this is important because this is where the pellets, this is where our sperm cells, if present, will be. And this is what our, what we're going to take a sample to make our slide from. So the next step is, we have our pellet of cells. We just need to take about half the volume of what we call the supernatant, so the liquid at the top, and remove that to another Eppendorf. So we need to use our pipettes. So open our pipette box. We use a P1000, which is 1mm pipette for this. Put the appropriate tip. Very careful. open the lid and remove approximately half of the liquid and leaving about 0.1 mil left in the actual tube, and transfer into another Eppendorf. Get rid of your tips appropriately. So now we're left with about 0.1 mil with our pellet. We need to resuspend this pellet, and what that word means is you need to mix the pellet back into solution. And how you do that, you get a small pipette volume, probably about P200 pipette, and you want to kind of like put your pipette tip in the solution and up and down with your thumb till you can see your pellet go into suspension. Once that's done, you want to take a sample of that, approximately 30 microlitres, and apply this into the centre of your microscope slide. So you might want to put your microscope slide down, and very careful, 30 microlitres into the centre of your glass microscope slide. Carefully keeping it horizontal, transfer this to your hot plate. So you want to put your hot plate on around about just between 50 and 100 degrees Celsius. Health and safety here, it can get very hot, so handle with caution. And what's happening now, your resuspended pellet, a 30 microlitre sample has been put on here, and the purpose of the hot plate is to get the cells to adhere to the glass microscope slide. And how you monitor this, there's no set time. You have to kind of observe the pellet, the cells, just kind of evaporating. And until it goes like a white colour, you know that has now stuck to your microscope slide. So this could take between 2 minutes or 3-4 minutes, depending on the sample really. So it dries from the outside inwards, and then once you can see all the solution has been evaporated, you are then able to go to the next stage. So once it's dried, you can see here, be careful when you handle these slides, it can be warm, but just be very careful, and you can see now it's gone white and it's basically stuck to our microscope slide. We have a washing up bowl here with a rail, and this rail, you can sit your microscope slide onto this rail. We have two stains here called hematoxylin and eosin. The first stain we use is hematoxylin, the purple kind of looking stain here. Using the dropper bottle and having a stopwatch quite close to you, you need to set this for 2 minutes. Using your dropper bottle, place 2 or 3 or 4, there's no quantity there, 2 or 3 drops. Set your timer for 2 minutes, and the hematoxylin, if there is sperm cells present, stains the sperm head in like a two-tone kind of colour reaction. And that's what we have to do. You have to leave this for a full 2 minutes. While that's happening, what you need to do is prepare and have some distilled water on hand ready. Because after the 2 minute time period, you need to gently wash the Harris hematoxylin stain from our glass slide. You might want to do some tests, because if you press too hard, you pretty much power wash your sample from your slide. You want to kind of get a nice gentle flow of water ready for when your 2 minute time period is up. So once your 2 minute period is, you want to kind of hold your slide up face down, let the solution go into the washing up bowl. Nice gentle wash. Again, please don't press it too hard and power jet it. Just nice Nice gentle kind of wash. Wash the excess off. Place this back onto our hot plate, and you just want to evaporate the water from your sample. This may only take about 10 seconds. Caution here, only do 10 seconds because you don't want to leave it on too long because you will dehydrate your slide sample. So once the water has been removed, place this back onto your rail. And this is now where we use the eosin, which is the red-orange looking solution. Using your dropper bottle, you need to set your timer for 30 seconds here. So 2-3 drops of eosin, press go, and you want to do this for 30 seconds. Again, get your water ready on standby. And I'm just counting up here, so it's reached 30 seconds. Now, again, holding your slide down, nice gentle washes. The eosin stains the sperm tail. So now you have 2 stains on there. The hematoxylin staining the sperm head and the eosin staining the sperm tail. This now goes for the final time onto our hot plate just to evaporate the excess water. Again, should only take 10 seconds. Brilliant. So what you have now is your specimen, which is 30 microlitres from your sample, from the swabs, from the underwear. And on here you can see that you have stained using 2 stainings, and you can see your specimen, which has now appeared. You can see it through the glass. What we have to do now is apply a cover slip over our specimen to protect it from anything landing on there. And to do that, we use a solution called Histomount, which is kind of like a gloopy, syrupy, transparent solution. And we need a small glass slide, which is a very small, square piece of, very thin piece of glass. So be very careful when handling this and get one ready. So sometimes the bottle can be hard to open, and you have like these dropper bottles, and as you pull it out you'll see this like a transparent, gloopy kind of solution. With care, you want to kind of hover over your sample and let gravity do the work. You may have to go back and get some more if you haven't got enough of the original. Try not put too much on there. It's just by eye really, of let gravity do its work. And once you have like a very small kind of blob of it, you then get your cover slip, your little small square piece of glass, and at a 45-degree angle just let go of it over your solution and let it nicely fall on top of your sample. And you'll see gravity pushing the cover slip down with a very small pinch. Just squeeze it down, and what happens there, you've got a nice cover over your specimen, and you are now ready to view this under the high power microscope. So the next stage is we have to see what's on our microscope slide. So we've made our H&E slide, which is a hematoxylin and eosin slide, and we use a high power microscope. So this microscope's the high power microscope because they have high magnification. So these can go up to, in this case, times 400 magnification. So we have our slide and we have this black area here called the stage. We place our glass slide on our stage and we always start off on the lowest objective. So these colourful kind of cylindrical kind of shapes are called our objectives. And we turn the microscope on here on the side, and we got times 4 times 10, which is times 40 magnification. This little section here adjusts our stage. It moves the stage around, so we're able to search our slide. This section here, these two kind of adjustable kind of circle kind of objectives here are our fine focus. So we first adjust the eyepieces to fit yourself, so you might have to move them up and down so you are comfortable. And on the left is your fine focus, and then you search the slide to see if we can actually see any cellular material, and secondly, in this case, we'll want to look to see if we can see any sperm cells. Once you've got it in focus, you may want to adjust your objectives to go even more magnification. So in this case, it's times 40, and these eyepieces are times 10, so that's 40 times 10, 400 magnification. You want to adjust your fine focus, and you want to search your slide. You can either go left to right or up or down until the whole surface area is completed. And if you see anything of interest, you want to write it and update your notes. A normal slide could take you 20 minutes to examine. Thank you for listening to the tutorials today. If you've got any questions about any technical detail or you'd like to speak to us, please contact the module leader, myself, Paul Langton, either on email or Teams, or just pop in, knock on the door and come and see me.